in about a minute and then we'll get started. So we are happy to have with us today Dr. Mohammad Rizwan from Toronto. He will be sharing with us his work he is doing on biomaterials, where he is using micro and nanotechnology for tissue regeneration, which is the idea of regrowing and replacing diseased organs and tissues. He's a graduate from University of Punjab with bachelor's. Then he did his master's from South Korea in bio-nano engineering, and he got his PhD in biomedical engineering from National University of Singapore. He has been a postdoctoral research fellow in Canada for a few years, and I know he's doing some fantastic work. This webinar is hosted by STEM Matters. So STEM Matters is a nonprofit registered here in USA. It is formed to inspire students from high school all the way to undergrad and graduate in areas of science, technology, engineering, and environment matters. We try to bring speakers who can nurture critical and analytical thinking skills and make us learn from them and possibly inspire to do similar work. STEM Matters engages, guides, and enables students from all over the world to innovate, invent, and deliver high-quality solutions to challenges of our times. We have developed many research and educational programs to provide local, regional, national, and global activities to learn from each other, to collaborate with each other, and possibly to compete for good outcomes, excellent outcomes. So let's get started, and over to you, Dr. Rizwan.
Thank you very much for inviting, and thank you very much for the nice introduction, Dr. Iqbal. So really excited to be here. I will be sharing my research on basically developing biomaterials that can be used for growing either new tissues or for replacing the diseased tissues, as Dr. Iqbal mentioned. And you'll see that gradually, like a transition from more engineering towards more cell biology. But I understand that engineers are usually turned off by cell biology, but believe me, it's not that difficult. It's really exciting research, as you will see, and it really directly impacts healthcare. I mean, we all want healthcare to be improved, and we want new innovations to be incorporated into the healthcare system. So I'll share two stories today. One is related to the blindness, and one is related to the liver. Okay. So tissue engineering, it's the idea of tissue engineering really is growing tissues and organs in lab for patient treatment. Let me just turn on my pointer if I can. So, okay. So in the Scientific American in 1999 basically presented this idea in a series of articles saying that we'll have a brave new world in which people will not need to die just for the lack of spare parts. And by spare parts, really they meant that these tissues, let's say if you need a liver tissue or if you need a new heart, right? So instead of waiting for a donor, we can basically generate that in lab. So the cycle really how it goes is we can get stem cells from human, let's say. So stem cells are cells which can become any other cell type. So they can become skin cells, they can become heart cells, they can become brain cells. So there's a lot of biology involved. So we extract these stem cells from humans, we expand these cells, and we convert these cells to our desired cell type. If we are working with, let's say, regenerating a new heart, so we'll convert stem cells to heart tissue. But in that conversion and in that tissue engineering, we really need biomaterials that can direct these processes. This is where the engineering comes into tissue engineering. Without engineering, basically, it's not really possible to develop new tissues. This is where we basically marry biology and engineering or material science to make these tissues. So once we have basically developed these tissues, then we implant these tissues in the humans who are in need of these tissues. If, for example, if you are waiting for a transplant or a donor, it can really take several years for you to get that. But the idea is that we can generate tissues in probably in few months or even faster for you to receive those implants. So this is basically the idea of tissue engineering, making new tissues in lab by using engineering principles and by using cell biology principles. So before I delve further, I just want to basically introduce the concept of a hydrogel. Like a hydrogel is the most commonly used biomaterial in tissue engineering. And why? Because it really resembles human tissues. I mean, we are mostly made up of water. And hydrogel is also mostly made up of water. It's a water-soluble cross-linked polymer network. And I'm sure many of you have seen jello or jelly, right? So jelly is a hydrogel. It's really that simple. So, but once we, like jelly is not stable, but once we introduce extra crosslinks, we can make it a permanent hydrogel, which will stay there. Here you can see that the hydrogel can be engineered to make this really thin film, which I used for cornea, as I'll explain later. We can also nano pattern this hydrogel just to control cell functions on these hydrogels. So hydrogel is developing tissue engineering. Okay. Now, so we have a hydrogel, and really the goal of a biomaterial engineer is to control cell functions using a hydrogel, right? So we can have different types of cells, right? When we put these cells in a hydrogel, we want to control their functions, like how they are growing. We can grow these cells by changing or by tuning the properties of these hydrogels or mechanics of the hydrogel. We can differentiate cells in these hydrogels to make other cell types, right? You can start with stem cells. You can get brain cells by just playing with the material properties of these hydrogels. And you can also induce self-assembly. By self-assembly I mean that you introduce these single cells and they ended up making these beautiful vascular structures or bile duct structures in this case. How does this happen? It really is a lot of engineering involved in making these hydrogels, and they can ultimately basically control how cells are growing and how cells come together to form these structures. So how do we control these things? Like how do we control cell functions in a hydrogel? It really, there are three things we can do. One is we can have micro-nano topography on these hydrogel surface. For example, here you can see that there's really a jelly or a hydrogel that has these micro-nano features, and it has been shown, and I have shown that we can really control how fast cells grow and what their functions are, depending on what kind of micro and nano features are there on the surface of the biomaterial. Then we can have biochemical signals, right? So during development, all these organs that we have in the body, they really come from a single cell. And there's a lot of signaling involved in these cells. And those signals are biochemical signals coming from other cells or coming from the environment itself. So we can have these biochemical signals that can be basically engineered into these hydrogels using chemical engineering or chemistry. And then we can have mechanical properties of the hydrogel. So mechanical properties can basically tune, again, how cells are functioning. You can have soft hydrogels, you can have very stiff hydrogels, and depending on their stiffness, the cell functions will be completely different. And I'll really show today one related to cornea, and the second story is related to liver, that how we use these principles in biomaterial design to grow these corneal tissues or liver tissues that we can transplant in the humans. Okay, so I'll start with the cornea. So like cornea is really the front transparent part of the eye. And this transparent part of the eye, when we go deeper, we see that it's a layered structure. Like the bulk of the cornea is something called stroma. You don't really need to pay attention here. What I really want you to pay attention is the endothelium. This is just like a layer of cells in the posterior corneal region. So these cells are very important, even though it's just a layer of cells, but they really help cornea or our eye to maintain its clarity and transparency so that we can actually see. So what happens when these cells are damaged? So remember, cornea is world's most transplanted tissue. Like it surpasses really any other tissue transplantation. And 40% of the transplants are really due to that damage of one cell layer that happens in the cornea. And currently, I mean, just to give you an idea of the severity of the problem, 53% of the population does not have any access to cornea. Just because we have a limited number of donors and the disease are becoming more prevalent as we are getting older, the blindness issues are becoming more severe, and we don't have that many donors. So we really need new approaches to make these cornea or these tissues in lab so that we can treat patients. So what really happens to this layer? Something really happens in the back of the cornea, on the posterior side of the cornea, that we start to see this micro features on the corneal surface. And that really leads to the corneal blindness. We see that the cornea has become really opaque, and which affects the vision. Or in other words, what I'm saying here in the schematic, that on the back side of the cornea, in a normal cornea, everything looks great. We have a nice monolayer of cells there. But when you get that disease called Fuchs dystrophy, in that disease, you start to see these micro features, and that ultimately leads to the death of these cells. And once these cells die, cornea becomes cloudy or non-transparent. So let's say we have, or someone has this disease, right? So what are the options? How can we treat these patients? Really, the only option that we have now is we take this cornea from a donor, and then we transplant that cornea into the patient. So basically, we remove the patient cornea and we put this cornea into the patient eye, and we suture this. Of course, it's a painful procedure. It takes several months for full recovery. But there's another problem. We can only treat one patient with one cornea, right? But as I mentioned, that we don't really have a lot many patients or a lot many donors. So what is the tissue engineering approach? How can we treat more patients from one cornea? Well, what we can do is we can remove these cells. And after removing these cells in lab, we can grow them. Remember, I mentioned that by using biomaterials, we can control their growth, or by using cell biology principles, we can control their growth. So if we can control their growth, now we can basically have a lot more cells that are coming from the same one cornea. And after we have grown all these cells, we can basically put them on a hydrogel film to generate a tissue engineered graft. And then this graft can be transplanted into the eye. Now, because there is a lot of cell growth involved, so we can treat up to 20 patients from the same one donor cornea. So you can see that how basically we can use these tissue engineering principles to ideally treat many more patients instead of having one donor to one patient. Now, as you can imagine, it's not that simple, right? So to generate this tissue engineered graft, we really need biomaterial scientists who can develop this hydrogel film that is going into the eye. So what is the design criteria of a hydrogel that is going into the eye as a carrier for this cell layer? Well, it has to be transparent, right? It's going into the eye. It should be non-toxic. It should be slowly biodegradable. I'll get into this later. And it should be very strong, right? Hydrogel or jelly, if you remember, right? It's not very strong. Here we are talking about a thin film in which we have cells, and surgeons or corneal surgeons, they want to transplant that into the eye. So we really want to make sure that during all these procedures or surgeries, it does not really break. So we need some engineering there to make it stronger. And then of course it should support the the monolayer formation, right, on this hydrogel. So with that design criteria in mind, now we can move towards developing a hydrogel that can have all these functions. So I opted for a hydrogel really based on jelly. The jelly is mostly based on gelatin. So I started with gelatin for a number of reasons. It is a biopolymer, cells usually like it, cells usually attach to it, and it can be degraded. So there are a number of functions already there. So if we can do some modification, some chemical modification to make gelatin methacrylate. Why? Because methacrylate groups, they can be cross-linked using UV light. So basically we can shine UV light on this hydrogel, on this solution, methacrylate solution, and it will form a nice hydrogel. So, I mean, the idea is simple. We start with gelatin solution. We do some modification to make gelatin methacrylate. We shine UV light and it'll form a gel. This gel can be formed into a thin film or can be formed into, I mean, a bulk 3D culture hydrogel also. But there is a problem. The problem is that it is mechanically weak, especially when we make a thin film hydrogel. It really tends to break. When surgeons want to transplant into the eye, it'll just break. And then it degrades really fast. So we want to slow down the degradation. And lastly, we want to further improve it so that we can, when we put cells on this gel, they really like this gel, and we can really grow a monolayer, a transplantable grade monolayer on this. So how did we solve these challenges? So before that, like, my question is, why gels in fridge, right? So if you have made jelly, you know that we heat up this solution, and then this solution really, we put into the fridge and it gels. Why does it gel? It gels because gelatin, it forms triple helical structures when we cool it down. So those random chains of gelatin molecule, they will basically get together to form these triple helical structures. And this non-covalent interaction between different chains, it will cause it to gel. And that's how we get a jelly. And actually, if you heat this jelly, it will become a solution again. Because it's non-covalent, it's non-permanent. So I thought, can we combine two types of cross-linking in the hydrogel to make it stronger? Can we have thermal gelation, in which we basically cool down the solution first, and then we can have UV curing, or when we shine UV light on it, it'll probably give us a stronger gel because we are having this double cross-linking or sequential cross-linking in this gel. So with this hypothesis, we tested the properties of this hydrogel, and we learned that the GelMA hydrogel is significantly stronger. If you compare that, the GelMA Plus, we call the new hydrogel as GelMA Plus just because it has this dual or double cross-linking. So you see that when we increase the concentration of the hydrogel, the GelMA Plus is really stronger. It's up to 5 times stronger than a regular hydrogel, just because we introduced that extra cross-linking step or extra incubation step at 4 degrees centigrade. And then most importantly, when we bring it to 37 degrees centigrade temperature, we see that the strength really is maintained. It does not really go away. So GelMA Plus really holds its strength compared to a regular hydrogel, or regular gelatin hydrogel. So this was great. It really solved first challenge, that now we can make a thin film, and surgeons are really able to implant that film into the eye. So after solving that challenge, we also wanted to make sure that we understand, like, scientifically what's really happening in this hydrogel. Like, why it does not lead to lower strength when we heat it. So when we know that when gelatin turns to jelly, these chains become these helical structures. This is an exothermic process. And jelly to gelatin, like when we heat it, it is an endothermic process when these helical structures become these random chains again. So we use differential scanning calorimetry, but just to see what's really happening in these gels. And it showed that these red lines, which is the hydrogel, it does not have that thermal transition curve, but the solution does have that thermal transition curve. It means that those triple helical structures which we formed in this hydrogel, they really become permanent. And that's why we don't really lose any strength when we heat it to a higher temperature. Okay, so my previous research had shown that if we take these corneal cells and if we basically culture them or put them on these biomaterials with different micro or nano features on them, we can really control how cells are growing. So we can have, let's say, 250 nanometer pillar structures on a biomaterial surface. In this case, it's a polystyrene simple cell culture where we can have wells, we can have pillars of 1 micron diameter. And what we noticed that when we compare to a regular flat surface, these 1 micron pillars were really giving us a better cells. These green lines that are appearing at the boundary of cells, they really are supposed to be there when cells are of good quality or transplantable grade quality. And this, the expression of that Z1 protein, it was less. I mean, in summary, I don't want to get it more complicated, but the idea was that 1 micron pillars can really improve the quality of cells. Now, the question is, can we pattern our hydrogel surface with these micro-nano features so that we can further improve their quality when we are basically putting it into patient eye? But I mean, patterning on a hydrogel surface is more challenging compared to plastics. But we developed a new method of Nanoimprint lithography that we can use basically to pattern hydrogel surfaces also with really nice features. So here you see a really thin film hydrogel patterned with 1 micron wells. Here you can see again 1 micron pillars patterned on a hydrogel film, and we can really go down to 250 nanometer scale on the hydrogel surface also. You can just see here the hydrogel films patterned with these nano or micro features, and we can actually pattern multiple topographies on the same chip, right? Sometimes we want to identify which pattern will affect cells in what ways. So we want to really have a high throughput system where we can have a number of topographies on the same hydrogel chip so that we can study their effect on cells. So after we achieved this, then we looked at how it really affects cells. So you see, same hydrogel, when we don't have those pillar structures on them, the cells again are not really showing or not expressing that green protein shown here strongly. But when we have 1 micron pillars, the expression of that protein is really high, which was good, right? As I mentioned that we want to improve the quality of these cells. So we basically engineered this graft with these cells on it, and then we also tested the glucose transport. So one of the function of cornea or these cells in cornea is that they provide glucose to the rest of the cornea as a food so that cornea basically stays healthy and stays transparent. So we want to make sure that the tissue engineered cornea endothelial graft that we are making in the lab, it can also do the same function. And it showed that really it can diffuse, it can basically, I mean, glucose can be transported from these hydrogels. So in the quality wise, it looks great. The strength was great. And we were really ready to transplant that into the animal models, right? So how do we transplant this in animals? So for that, we really have a device developed in Singapore. And this device, let me see if I can, it's probably, let me see. Okay, so this is really the device that we can use to transplant into the eye. Can someone confirm if you can listen to the sound? Okay, good. So this is, you can consider that this is a hydrogel with that corneal cell layer on top that is going to be into the eye.
And a small amount of dispersive viscoelastic is added. A capsule glide designed to fit through a 4 millimeter incision and to accommodate a donor measuring up to 10 millimeters in diameter and 250 microns in thickness is locked into position upside down onto the perforation base. Forceps are introduced through the glide to grasp the stromal edge of the posterior lenticule. coil configuration with no endothelium surface.
So this is where I mentioned that we really need strength in the hydrogel, right? Because we are doing all these manipulations. And if the strength is not there, it will simply break during these procedures.
Is touching. A glide introducer is then attached to the back end of the capsule. The assembled endoglide is removed from the base, inverted the right way up, and is ready for insertion. With an AC maintainer on to stabilize the chamber, the endoglide is fully inserted. The 4mm sclerotunnel forming a complete seal of the wound. Descent forceps introduced from a nasoparacentesis then simply grasps the donor and pulls it in. The donor automatically uncoils and the endoglide is removed. A small air bubble is then injected beneath the donor to prevent descent onto the iris.
Okay, so this is how we basically transplanted the grafts that we engineered in the lab into the eyes. So what do we see? We see that once this is the hydrogel film into the eye, and this is the original cornea of the animal model. So these surgeries were done in rabbits. So we studied that for 4 months, and after 4 months, we could see that it's slowly degrading because these pores are becoming bigger compared to those gels which were not put into the eye. So the idea here is that after we transplant cells into the eye, this hydrogel film will slowly dissolve, and ultimately we will just have cells which are attaching to the cornea. And this is how we basically we will end up, reversing the blindness issue. Now, we also want to make sure that the hydrogel is biocompatible. So we put these hydrogels and we observed rabbit eyes for up to 4 months. And we really did not notice any negative influence or negative effects in the eye. So it showed that here, this arrow is pointing towards the hydrogel film that is transplanted into these eyes. And you can see that really the eyes remained clear and there was no negative consequences of putting this biomaterial into the eye. So that shows that this is a biocompatible system of generating this graft. Okay, so is there any question so far? I'm just changing gears slightly. So there's no question as a simple question. So just ask me anything, like even if it's a naive question, completely fine.
So I got a question from one of the students. He just asked me that what happens to the donors? So my understanding is that the corneas come from people who donate during their lifetime, but we take it when they die, right?
Yeah. Oh, yeah. Good question. So yeah, of course. So the donors are basically those who have opted to donate their corneas after they have died, not in their not in their lifetime, right? So yeah. So these corneas come from donors who have died, but of course they are preserved and they are used like within a couple of days. So basically, we were getting donor corneas from patients in Singapore, and we were removing cells, and then we were growing those cells in lab so that we can generate more grafts. Yeah. Okay.
There is one more question.
Yeah.
What about the sterilization? Can you elaborate on this?
Yeah, absolutely. So great question. So I have actually, so these hydrogel films, these were sterilized by using simple filtration. So we basically in the labs, we have filters which can remove the bacterias or the contaminations. But I also published a study in which we compared different sterilization techniques. So because this is related to healthcare, so these things are really tightly regulated. So for example, in USA, FDA basically have guidelines of sterilization of if it's a device or if it's a tissue, how should it be sterilized, or if it's a hydrogel. So we compare different methods of sterilization, and we found that really, autoclaving is one type of sterilization that worked well, but I wouldn't want to go into this rabbit hole. So sterilization can also affect mechanical properties of the hydrogel. So it's a good question. Yeah. But this was sterilized by using simple filtration. Okay, so I'll move forward. Feel free to ask questions, even in this section later. Yeah, so I mentioned that when we get this disease, patients usually have these microstructures on the back of the cornea, right? This is an image of a 36-year-old patient, which is an early stage of the disease. We can see that there are these few micron tall and wide structures onto the cornea, right? So there is another line of research which says that instead of generating these tissue engineered grafts on a hydrogel, we can simply grow these cells in lab and we can inject these cells into the patient eye. And after we inject these cells, we ask basically patient to lie down for a few hours so that cells attach to the cornea and cells will form a new monolayer into the cornea. So sounds great, right? So we can basically simply, we don't really need a hydrogel. We don't really need to do all that engineering. We can simply do cell therapy. We can just inject cells into the eyes. So my question was, we know that in the patients, we have these microfeatures on the back of the cornea. And when we are injecting these cells, how will these microfeatures affect the regeneration, affect that monolayer regeneration or formation? Like, will they interfere with the cells? So with that question in mind, we proceeded to basically develop a system in which we can study this. Again, there's a lot of microengineering involved here. So we created, using these microfabrication techniques, we created features that can basically resemble different stage of the disease. I mentioned that in the early stage of the disease, we really have these curvy-like features or lens-like structures, but as the disease progresses, we can get taller and taller features on the cornea. So we basically developed this method which allowed us to replicate different stage of the disease so that we can test them in terms of cell therapy approach. So with that, we looked at 3 different features which were like 20 micron height, which represented the late stage of the disease. And when we put cells on these features, we saw that cells were mostly dying, like all these cells on top of these structures, they are dead cells, and they were not really forming that nice monolayer. Like there was a lot of cell-free areas. In the middle stage of the disease, when we basically reduce the height of this, now we start to see that even on top of cells, we can have that green lines, right? Which remember I showed you earlier that represent the quality of these cells or the monolayers. And when we further reduce this height and change it to this curved structure, that really represents the early stage of the disease. Now we got the best results here, that we have an almost complete monolayer formation. What it really shows us that cell therapy may not work for every patient. If it's an early stage of the disease, we might be successful in just basically injecting cells and they can form monolayer. But if it's already a late stage or middle stage of the disease, we probably need a tissue engineered graft in which we can control the quality of the graft and then we can transplant that. So this was really the first example that we were able to study this disease in a dish, and we could conclude that we can basically correlate the stage of the disease with the cell therapy approach. And just to basically really prove this message or show this message, you see when we don't have anything on the surface, this really represents a normal cornea. And you see cells are basically growing on this surface and really making this monolayer. So the shape of the cells is like more polygonal and it looks like a monolayer structure. But when we have a surface with these microstructures, which represents a diseased cornea, now cells look completely different. They are more elongated, they are really confused, they don't really need to know, they really, not really making a monolayer. So even after 24 hours, you can see that the difference between these two surfaces in terms of how cells are behaving or migrating on this surface. So yeah, really, it's really important to consider the stage of the disease for cell therapy approach. Okay, so in the second, probably in the next like 15, 20 minutes, I'll talk about the liver. So this is a more recent work and really exciting work that we have been doing in liver stage. So with cornea work, it's still ongoing. We are still collaborating with the surgeons in Singapore, and they are now testing like more advanced things. For example, how long does it take to reverse the blindness? What is the strength that what is the thickness of the graft that we need? How many cells should be there in each graft? So some of these things are still ongoing, and we hope that probably within a couple of years, they should be able to start clinical trials in Singapore with these grafts. So moving towards liver. Now, liver is another like tissue in which really affects the quality of life. There's like more than 500 million people are affected by liver diseases. So just to remind you about the anatomy. So liver is really made up of, there are many different cell types, but you can consider for the purpose of today's talk, there are two really important cell types. One is this red called the hepatocytes. You don't need to remember, but really I want you to focus on these green channels or these green tubular structures in the liver. These are called bile ducts. These bile ducts, as you can see in the inset here, they really basically collect the bile, which is produced by liver, and then they transport that bile. Basically, it's like highways to collect all these bile acids and then transport to the gallbladder. And they really have a very important role. For example, 66% of the children or these juvenile liver transplants, they are caused by diseases which really target these bile ducts here. So my research was really focused on basically engineering these organoids of these bile ducts to see how we can get cells from stem cells to basically regrow these bile ducts in the patients, or we can make organoids that can be used to transplant into the liver so they can regrow these bile duct structures in the liver. So there are a number of options when we think of liver in terms of treatment. So we can do liver transplantation. Again, the classic, gold standard that we can have a donor who can provide liver tissue. But again, there's a really long waiting list if you are on a waiting list for liver transplant. The other option is cell therapy. So we can have stem cells that can be converted to liver cells, and then these cells can be transplanted or injected into the liver to basically hopefully regrow the liver function or liver and restore their functions. The third option is really the liver dialysis. Just like kidney dialysis, we can have these devices that can basically filter or purify the drugs. And the last option is the tissue engineering based option. In the tissue engineering option, we can basically grow livers in lab by combining cells and biomaterials. So I'll quickly talk about cell therapy, and then I'll move towards the tissue engineering part. Again, so this part is about stem cells, or really just to remind, the stem cells are cells that can be converted to different types of cells. We can basically transform or convert stem cells to muscle cells, to blood cells. We can make brain cells from these cells, heart cells, liver cells. So today I'll be focusing on basically this aspect of converting stem cells to liver cells. So in our collaborators here in Toronto, they really developed a new method in 2015, which could be used to convert stem cells to liver cells. But it was a mid stage of liver, or or more appropriately called progenitor cells. So this mid stage liver cells, then they put these cells on top of mouse cells as a feeder layer. And these mouse cells were sending signals to these mid-stage liver cells so that they can become liver bile duct cells. Now, as you can imagine, this was a really critical step, that putting these cells on mouse cells. The question is, will you receive this cell injection for your treatment, which you know that they have been cultured in contact with mouse cells? So really, these cells are not appropriate for cell therapy. Of course, they are really useful for a number of different applications, but for cell therapy, we really don't really know. Even when we are able to isolate mouse cells, of course, we can purify them, but we are still not sure what was secreted by mouse cells and how will they affect the properties of these cells going forward. So our question was, can we engineer a hydrogel? Really, so we are all about engineering biomaterials. So as an engineer, so our question was, can we engineer a hydrogel that can really replace these mouse cells to differentiate or make these liver bile duct cells? So with that motivation, what we really did, again, there's a lot of chemical engineering and chemistry involved, which I'm skipping, but feel free to ask if you have questions on the chemistry side. But the idea here was that we have stem cells that are differentiated towards this mid-stage liver cells, and then these cells are put into this engineered hydrogel. The beauty of this hydrogel was that it can provide signaling just like mouse cells to convert cells to cholangiocyte cells. So basically we were replacing those mouse cells with our synthetically defined hydrogel. So how did this work? We noticed that when we basically culture these mid-stage liver cells on these engineered hydrogels, which can provide not signaling or the same signaling that mouse cells were providing, we can actually make beautiful bile duct cells compared to the HA hydrogel, for example, in which you don't really see those markers. These markers are really important. I'm just showing two markers. Markers are just characteristics of cells, which we check when we are converting one cell type to another cell type. So we check these markers and we saw that cells have actually become cholangiocytes, and not just markers. We also saw these morphological changes. Remember, I mentioned to you that these bile duct cells are really making those tubular structures in the liver. So, and we saw that even in this 2D or on the top of gels, when we provide them these chemical signals, they were trying to make those tubular structures on the hydrogel surface, which was not happening on the regular hydrogel or without those signals. So this was good to see that we basically provided a way to improve cell therapy for patients, hopefully in the future, that now we don't really need to mix mouse cells with the human cells to convert them to liver cells. And because we were using those chemical engineering approaches, so we can really have a nice control of how cells are growing and where cells are differentiating. So we can have photochemical approach, and we showed that we can actually turn on and turn off those signals by simply shining light on them. So when we shine UV light on it, we can basically initiate or stimulate that signaling that converts cells towards a different lineage. But when we don't shine UV light, the protein signal is still off and cells are not differentiated. And we can actually also do this in 3D. I mean, we have not differentiated in 3D, but we have already shown that we can selectively pattern those signals inside the hydrogel in defined regions by using laser in different locations in the hydrogel. This is very critical because if we are really thinking about making a liver in which we have different types of cells, we want to control which cell is being differentiated. So now really, I mean, last few slides, I'll talk about.
Dr. Rizwan, we have a few questions also that have come up. So I don't know if you want to address them while we are going.
Yeah, absolutely, sure. Yeah, that'll be great actually here.
So the most recent one is, can you please tell the source of this hydrogel? From where are you obtaining the gelatin?
Okay, great. So yeah, so I should have mentioned. So first part, the Cornea project, we used gelatin hydrogel. The gelatin hydrogel is, so there are a number of basically options. We can get them from bovine. We can also get them from porcine. So we use the hydrogel that was basically extracted from bovine. And in the case of this liver project, we are using a hydrogel, which is, again, so our, like the selection of material should be really dictated by the biology, right? So in this case, we are using hyaluronan hydrogel. So hyaluronan is really, I mean, if hyaluronan is used for skin, so I know that there are a number of products based on hyaluronan for skincare, but hyaluronan really comes from, you can see that I mean, there are a number of basically ways we can get hyaluronan. Mostly comes from microorganisms, so it's more well controlled. We can basically get them from bacteria. We can also get them from algae. So there are a number of ways basically to make hyaluronan. Yeah. So different materials. for different projects.
Right. Very good. So another question is from how much advancements have been made in artificial human organs using advanced biomaterials and stem cell technology?
Yeah, so I mean, this is the question, right? We are trying to answer here. So I am presenting two advancements here that my research has shown. One is on cornea, and one is in the liver that I'm talking about. Like, if you want me to give an overarching view, I mean, I would say that a number of advancements have made, but we are still not there yet. Like tissue engineering promise is to provide tissues for transplantation, right? And stem cell discoveries have, again, there's a lot of progress has been made. It really depends on tissue. In the case of skin, we can generate skin tissue, like tissue engineered skin, which is live. I mean, we have already succeeded in making that. There are a number of companies who are basically commercializing that tissue engineered skin. The second is cornea. There's a lot of advancements have been made in cornea tissue engineering also. But in terms of bigger tissues, for example, liver organ or heart or brain, a lot of things have been understood already, but there are challenges. The biggest challenge or the holy grail of tissue engineering is providing blood vessels in these bigger tissues, right? So you can make these big tissues, but you want to keep these cells alive in these bigger tissues. So building a vascular network in these hydrogels is really the challenge, right? So nature has a really sophisticated way of, I mean, making all these beautiful organized organs and providing these blood vessels to keep them alive and functioning. But again, there's a lot of progress have been made on vascularization strategies also. But so far we have not been able to generate a truly functional tissue that can stay alive for years, for example. Yeah. But we are working. We need engineers, right? So there's a lot of strategies that are where we need basically engineers to advance these efforts.
So there's another question, Nazir. Oh, that was my question. So when we are talking about the micropillars and cell growth, so have you looked at any phenotypical changes in those cells if they are exposed to a plane surface versus a micropillar surface?
Yeah, great question. Okay, now we're getting into the nitty gritty. So again, I'm not showing all the data here. So we looked at the phenotypic changes, yes. So we looked at the Z1 expression, we looked at sodium potassium pumps expression. We also looked at two other markers, the EpCAM, E-cadherin, and we also looked at focal adhesions. So we want to understand also the mechanisms of why micropillars affect this way versus nanopillars, they are affecting in a different way. And when we don't have any pillars, we don't see these effects. So why is this happening? So basically we looked at focal adhesions. So focal adhesions are basically cells, they attach to the surface. So they make these tiny adhesions. So those protein complexes, they are really important in mechanosensitive signaling, or providing this mechanical signaling to the nucleus, and then in nucleus the transcription happens and that changes cell functions. So what we realized that those micro and nano patterns, they are really changing the formation, size, and turnover rate of focal adhesions, and that was really affecting how they were sensing their microenvironment. Yeah.
Very good. So that's where really micro and nano is interfering with the cellular behavior.
Absolutely, yeah.
Fantastic. There's another question, and I have two students who have raised their hands. We'll get to them. There is another question. Are there any other methods available for cell therapy other than injection? For example, oral. So,
interesting thought there. So I would say no. So oral cell therapy is really, you want to make sure that cells, I mean, you don't want to eat cells, right? So you really want to make sure that cells basically go where they are needed. And if you think of food, where does it go, right? So cells, if it's an oral, probably not. But I mean, when we are on this topic, so for drug delivery, there's a lot of efforts on basically oral controlled release drug delivery. So that there's a lot of potential in developing basically drug delivery systems there by using biomaterials. But for cell therapy, you really want to inject cells where they are needed. Like if they are needed in blood, you want to inject in blood vessels. If they are needed in liver, you really want to, inject cells, around the liver or in liver. Similarly for cardiac tissues, yeah.
All right, so go ahead. We have a question from Aditya. Go ahead, please.
Thank you very much. I'm sorry I raised my hand because I'm using a cell phone to follow this Zoom meeting. So my question is more or less similar with what have been asked by the moderator, I think. To my understand that for the hydrogel, the surface have been patterned by certain type of the form and distance of the pillars.
Yes.
Just to grow the cells. But in the contrary, when you try to simulate the surface of the normal and disease cornea, the cell growth better in the smooth surface.
Yeah.
So I think it's
great question.
Contradictory between the cornea surface, for example, of the patient and the hydrogel.
Yeah, absolutely. Yeah, great question. I'm really happy actually that you asked this question. So it kind of shows that, okay, so people are really following what I'm talking about. So to answer your question, there are two types of patterns. What we are really doing here is that we are trying to mimic the disease cornea and normal cornea, right? But even in the disease cornea, if you look at the scale, these are 20 micrometer wide pillars. And even if you look here, like if you look at the scale of the final, it's difficult to see. So this is 25 micron pillars. And this is actually, also 20, but we also studied 40. So we are talking about really big pillars. And so they are bigger than the size of cells. So it's not that each cell, under a cell, we don't really have any features there. Like, but when we are really talking about micro and nano topographies to control cell functions, we are mostly talking about something below 5 micron. Like 5 micron, 1 micron, submicron features. In that case, we have a number of features underneath the cell. What's the difference? The difference is that cells perceive those features in a completely different way. In this case, these features are really an obstruction towards the movement or migration of cells. They are not really there to control how cells are growing. It's like an obstruction for cells to form a monolayer, which is really the disease condition, right? But in terms of micro and nano topography, we call that cues because cells sense that in terms of making their decisions of how they want to grow. So it's really that, in short, it's really the difference in scale of these features. So when we have something under the cells or on which cells are adhering, and there are a number of features, they can affect the biomechanical signaling of cells. But in this case, they are more like an obstruction towards the movement of cells. And see like how they're growing, it's completely different. But again, we didn't really check. Maybe they will affect growth. But in this case, we were really interested in their migration and their formation of monolayer. Remember, I told you that in the cornea, they form a monolayer. So we were really interested in monolayer formation. But yeah, it's a great question. In short, it's a difference in scale. Yeah.
All right, very good. So we have another student, but we had another question before that, which is, Rizwan, great word. Which chemistry did you use to turn on or off your JAG1 signals, and for how much time did you expose to UV?
Okay, so we really have someone who really knows stuff here. so, so, okay, I'll go here. Okay, so in this case, so we basically identified, I mean, the detail is in my recent publication, Advanced Functional Materials. So what we did here is that JAG1 basically is a protein that sends signals to activate notch signaling. So we identified a new way of caging or hiding this protein so that others can also understand. And for that purpose, we used nitrobenzyl moiety. So nitrobenzyl is a group that can be cleaved when we shine UV light on it. So basically what we did is we attached a number of streptavidin molecules around this protein, and they were attached using nitrobenzyl. So now we have an umbrella of streptavidin molecules, which are basically covering this jagged, and they are attached using a photocleavable molecule. Now, only when we shine light, we basically remove all those streptavidins, and now protein is exposed to signal cells. But when we don't shine UV light, the protein is still covered by using those streptavidin molecules. So it's really a matter of using sterically bulky molecules that can create that umbrella to hide its functions. So we use nitrobenzyl moiety. So detailed method, we can definitely talk about later, but it's also in the Advanced Functional Materials paper I recently published.
All right. So we have some student, another student, Myra, please go ahead. Oh, sorry.
Thank you, sir.
Hold on.
Okay.
Yeah, go ahead.
Sir, can we use this hydrogel for vivo transplantation?
For in vivo transplantation?
Vivo transplantation? Tissue related.
Oh, tissue transplantation. Yeah, absolutely. I mean, we have done like cornea transplantation, right? So that is one example we were doing transplantation. In the case of liver, so our recent work in Toronto here currently involves, again, I still have like 3-4 slides to go. So it's really involved in making those liver tissues and then transplanting into the livers of animal models. In this case, we are using mouse models, and we want to then see the survival of that mouse. So of course, yeah. In fact, for this translational tissue engineering purpose, we always consider the transplantation aspect because we want our hydrogel to be transplantable. Ultimately, we want to transplant that, right? So unless we are working with drug testing or drug development. So mostly it's, yeah, translation is a key part.
Yeah, sir, because I read most of the time that currently none of the used hydrogels are used for vivo transplantation of tissues. So I just asked.
Yeah, I mean, you might be right in a sense that there is no commercially available product that, I mean, we can use to transplant in humans, but there are a number of studies, and this is a really active area of research of transplanting. Even in our lab, another group is working on injectable hydrogels, and they are always transplanted. So in one tissue engineering project, the team is working on spinal cord. So the hydrogel is injectable. Basically, we mix cells with it. We load this hydrogel with cells in a syringe, and we directly inject in the tissue side, for example, in the spinal cord or in the brain in the case of stroke. That is, I mean, right there, an example of transplantation using hydrogels. And there are a number of groups are also working here. That's a good question.
Oh, thank you, sir. Thank you.
So I'll continue. Let's continue. There are more questions, but we'll talk about them when you get to the.
Okay, great. Yeah. So, okay, I was talking about here. So one last message I want to give about developing biomaterials. I mean, as an engineer, so what's really important to consider when we are talking about making a different, biomaterial. So in the last example, I just mentioned, so far I showed liver 2D cell culture by using signaling, which is provided by a hydrogel. Now we want to do a 3D cell culture. So we want to take those cells, which we just converted to bile duct cells, and see if we can convert into tubular structures, right? So those single cells, we want to make sure that the environment or the hydrogel is conducive for them to come together and form these tubular structures. Again, there's a chemistry is mentioned here. we basically use click chemistry to make these hydrogels. And we have, again, that jagged protein, which is really critical in the context of liver. So jagged protein was also immobilized onto these hydrogels by using the protein G in jagged introduction, or not introduction, sorry, the affinity. So this was the goal, making these ductal structures or bile duct structures. How did we achieve that? Before that, I'll just mention that we need to look at the mechanical properties of organs. May sound a strange concept, but mechanical properties of organs are key for improved biomaterial design. If we look at the different tissues, the tissues are stress relaxing. What does this mean? It means that when you basically do a mechanical testing or compress a tissue and hold it there, most of tissues will have an ability to dissipate energy and relax that stress, because there's a lot of non-covalent cross-linking in these tissues or in the microenvironment. So they have this ability to dissipate that energy. And the level or the rate of dissipation depends on the tissue. For example, we can have a brain tissue, right? So you can have skin tissue, liver tissue, or brain. And the brain tissue, for example, relaxes its stress in less than 100 seconds, compared to a liver tissue that relaxes and have not even fully relaxed its stress even after 1,000 seconds. So it really depends on the tissue type. So when we are developing a biomaterial in which we want to grow those cells, right, liver cells or brain cells, we want to make sure that the mechanical properties of the hydrogel are mimicking the actual tissue. And hopefully that will lead to better outcomes or better growth of cells. So we were working with HA hydrogels, hyaluronan hydrogels, cross-linked using PEG. And when we basically looked at the mechanical properties of the liver, we looked at stress relaxation. Basically, we took liver tissue from mouse and we did mechanical testing, and we found that it does relax stress, but our hydrogel was not relaxing stress because it was like covalently cross-linked. So we included laminin. It's a protein which is mostly found in probably a number of tissues. And as we increase the amount of laminin, now we can see that our hydrogel behaves similarly as liver tissue. It was also relaxing stress, just like liver. So basically, we developed a liver-like hydrogel for the cell growth. Again, I'm skipping a lot of details here, but feel free to ask me questions. Okay, so we have developed this hydrogel, and when we combine this hydrogel and we do long-term 3D culture of bile duct cells in these hydrogels, we can really see the effect that the liver-like mechanical properties, even though we have liver-like mechanical properties, but when we don't have jagged signaling, we don't really see these tubular structures formation. So it's a synergistic effect of those biochemical signals and mechanical signals that then promote this bile duct formation. So we added single cells, and when we include jagged protein to provide those signals, again, remember those signals which were provided by mouse cells and liver-like mechanical properties, now we see these beautiful structures. And these are really 3D because we are putting cells inside the hydrogels. And this is just shows an animation of how 3D these structures are. So we generated these beautiful bile duct organoids in these hydrogels. Now, where we are going with this research further. So we have this bile duct organoids that are grown, but to actually generate a liver, which is a complex tissue, right? So we need more types of cells. So now we want to combine hepatocytes, another really important cell type in the liver, so that we actually get this beautiful complex liver tissue that can be tested in animal models, and ultimately, hopefully down the road, we can test in humans also to see if we can restore the functions in those patients in which liver is not functioning properly. But it's not just transplantation. I briefly mentioned that we can also use this tissue or these gels for drug testing. We can probably have better outcomes of drug testing if we are testing them into human cells which have already formed these tiny organoids. So ultimately, yeah, this is where we want to go. So in summary, I mean, really, I want to take 3 messages from this talk. Tissue engineering can really transform patient treatment, but we are not there yet, but really a lot of progress has been made in this area of research. We can learn from nature, like how how natural tissue development happens in the body, and then basically we can have improved biomaterial design from that. And lastly, again, we really need synergistic efforts of engineers and biologists to come together and work on this. I mean, just engineers cannot achieve this. Just simply biologists cannot achieve this. It's really a multidisciplinary field that really needs, I mean, synergistic efforts. With that, I'll just thank a number of people here. My advisors have been working in different labs, and a number of collaborators. Again, as I mentioned, that I mean, this is a multidisciplinary field. Really, a number of collaborators I've been involved in different projects in vascular tissue engineering, in corneal tissue engineering, in liver tissue engineering space that I've worked with. And finally, the funding sources in different countries, in Singapore and Canada, which I've been involved in different research areas. Yeah, so thank you very much. And now it's just time to ask. Please ask as many questions as you want to.
Thank you, Dr. Rizwan. Really appreciate this. Was an amazing talk. Fantastic. So, and of course, there are a few more questions. We have Emmett who has raised his hand. Emmett, please go ahead.
Thank you, sir. I'm audible?
Yeah, we can hear you.
Sir, I have a question related to replace or regrow the tissues with the bioaccumulated or nanoparticle used inside the human body as the potential to accumulate over a long period of time to reach a concentration that can cause the toxicity to the cell. So that it may be producing cancer or harmful effects or on the reproduction. So how can we indulge or deal with that? And the second question is that why there is no such a standard in the market to test that nanoparticles for human health?
Yeah, it's a great question. I know, I mean, even though I'm not directly involved in research, but again, I have been interacting with people in Cholangiocyte lab or in other labs who are working with nanoparticles here in Toronto. Yeah, you're right. I mean, the size of nanoparticles, like in nanotechnology, for example, the size is really important. The same material with the same chemistry can have different effect depending on the size of that particle. So some of that size cytotoxicity really comes down to, I mean, the size of that particle and how it's uptaken by cells and where it ends up and whether it's degradable or non-degradable. To answer your question, I mean, it's a great question, how we can deal with cytotoxicity. Really, I mean, probably the few ways in which people are tackling that is really the changing the properties or changing the size, or one method is basically coating those particles with something that is known to be non-cytotoxic. So there are a number of ways in which we can basically tackle that issue, either changing the surface properties of that particles. And then on the commercial testing, well, there is, I mean, because in the research and development phase, the systems are not really standardized yet. Every lab is basically trying to develop particles for different applications. I mean, it could be tissue engineering purpose. Mostly it's for drug delivery or for other therapeutic applications. So I guess when the field matures, only then we can have some standard, I mean, commercial systems available for testing cytotoxicity. So far, there are a number of different formulations that are being tested, number of different animal models that are being used for cytotoxicity. So yeah, we need to probably standardize.
Is there any standard yet for like for your lab to test the risk assessment for nano specified risks, sir?
In our case, really, we test, it depends on the tissue. So if you ask me the kind of the steps for testing cytotoxicity, if let's say if I'm working with liver cells, really the first step is to develop a hydrogel or nanoparticles if they are going for drug delivery in liver, and put those nanoparticles with the cells and see how cells are surviving. That is really the most important and critical test and key test to see the cytotoxicity. If cells are dying, even though your particles are great at releasing those drugs and the drugs are functional and you can tune the release rate, but you still cannot really use those particles. So in our lab, usually when it comes to nanoparticles, it's the cell-based assays, which are the most critical for our applications. One of the group in our lab is working with the double-dimensional method to make nanoparticles, PLGA-based nanoparticles, and those particles are being used for drug delivery in stroke models. So in the brain, basically, I mean, if we want to see the interaction of those particles with the neurons. So their first experiments are always making these nanoparticles, loading with drugs, and just in vitro, like in the lab, putting it onto cells or in the media and see if neurons or those brain cells are surviving or not. So if they are not surviving, it means we need to tune the chemistry of those particles. Yeah.
Thank you, sir.
There's
Thank
a
you.
related question here by another attendee is the hypersensitivity effects from the liver. So do you comment on that?
What exactly is the question?
So what are the hypersensitivity effects as it acting through liver? So I think this was probably a few slides ago. So hypersensitivity.
I mean, it might be related to the physiology of the liver.
Right.
Not exactly sure in the context of biomaterials.
Right.
The hypersensitivity. I mean, if you ask in terms of like if cells are hypersensitive to the materials, we luckily did not see that. We did not see any negative consequences on different cell types that we have tested on our hydrogels. But in terms of functioning liver, probably a medical doctor would be able to answer better instead of me.
There's another question about the commercialization there. What have been the hindrance or the challenges in commercialization of this idea?
Yeah, great. So that's great question. So exactly first challenge, it really came from that commercialization issue, this challenge. For cell therapies, so again, we are really fortunate that I'm working with Gordon Keller's lab, who is a really famous stem cell biologist in Toronto. They actually built a company called Blue Rock Therapeutics, which was purchased by another multinational company for like $400 million. Their whole research is, so Keller's lab's research is basically converting stem cells to different cell types and using those cells for cell therapy. In this case, they were limited that they had to use mouse cells. Again, so this is one of the issue for translation, right? So anything related to translation, you really need to identify every component that is in your system and characterize that. And with mouse cells, it becomes difficult because cells secrete a number of things in the media, which become very difficult to characterize. Versus a hydrogel, for a hydrogel, you know, right, what you needed and what chemicals were used to make that hydrogel. So you can easily track all those components and you can satisfy the regulatory requirements or translational requirements for the hydrogels. Yeah. So one of the challenge in the stem cell field, like if I give you a big picture, is for the differentiation, we use animal-derived products. One thing is called Matrigel. It's a hydrogel which comes from tumor to tumor, basically. Again, the use of Matrigel kind of limits the translational potential because it's a big challenge. Again, in Matrigel, it's difficult to characterize all the components. Yeah, so basically we want to make sure that in the system that we are developing is a synthetic system, or at least we know what's in there. Otherwise, it may take like years and years of characterization, and we may still not be able to fully satisfy the agencies or regulatory requirements. Yeah. And that's why engineering has a big role in the tissue engineering, right? Because biologists cannot develop these synthetic systems. So you need engineers basically to develop synthetic systems that can be used for biology.
Yeah. So that's the gap that we engineers fill, which is from discovery to technology.
Yeah, absolutely. Yeah.
How it can be really used. So I think there was a question about somewhere. Professor Malul asked about your email address. We have given him your Toronto email address.
Thank you.
But I can tell all of them that you can Google Dr. Muhammad Rizwan and you'll find his papers. They're well-cited papers, and you'll find his email address from there. And hopefully he will have his own lab very soon. So our best wishes are with him to become an independent PI, and we all will be looking forward to that.
Good.
All right, so if there are no questions, I think it's time to wrap up. Thank you, Dr. Rizwan. We really appreciate, and we really hope that we can get you to inspire many more of our students who are here and who are practicing these areas of research already. All right.
So thank you very much again for the opportunity. It's a really great audience. I mean, great questions, which I didn't expect. So happy to.
If you've seen the message, we have folks here from, of course, from US, UK, Indonesia, Tunisia, and I see some folks from Turkey as well. So we have the whole world covered here. So just good.
Great. Yeah, feel free to contact me if you have any